So now you know that proteoglycans are made up of a protein part and a carbohydrate moiety, that is GAGs. Now let's now move towards the biosynthetic pathway of these important compounds. These compounds, specifically the GAGs, as I have already mentioned, that they are made up of specific protein, specific sugar molecules. These carbohydrate moieties are either the amino sugars or the acidic sugars, and they are in alternative way. I mean, they are added to the molecule, to the GAGs, in the form of 2 units of 2 hexose molecules or 2 sugar molecules, with one molecule acts as the amino sugar while the other is the acidic sugar, and in this way these units alternate with each other. As we have already told you that these compounds are very important signaling molecules, they are very important structural molecules, they provide lubrication. So because of this very important, their importance, specifically the importance in this ECM, they occupy very special space in the field of biochemistry. Also keep in mind that in contrast to glycogen, which is synthesized in the cytoplasm, these molecules are synthesized in the Golgi bodies. So why in Golgi bodies? Because at the end of the day, either they become part of the cell membrane, or as a major part they will become the part of the ECM. So they have to be exported out. So they have to be then, for the exportation of these cellular molecules, they have to become part, they have to be modified in the Golgi bodies, and then they are enrooted towards the cell membrane and then become the part of the ECM. Before I move towards the biosynthesis, let's we revise a little bit about the structural aspects of proteoglycans. Although I have already showed you this, but I think you really need to focus a little bit more on it, so that whatever you will be studying in the succeeding topics, you will feel it more easy to understand those concepts. So the proteoglycan is composed of a core protein. The greenish part, the rod-like structure, is representing a protein, and then this is attached with 3 linker regions. These are the linker regions. These linker regions are actually 3 sugar molecules, like xylose, galactose, 2 galactose and 1 xylose, and they are connected with the serine residue, with the hydroxyl portion of the serine residue. Of course, the serine residue is the part of the protein, and they are attached with the serine residue, the linker region attached with the serine residue through the glycosidic linkage. This is ortho or O-glycosidic linkage. And then finally comes the repeating disaccharide units, which of course make the part of the GAGs. So in this way, this is the full major part of the structure of the proteoglycans. And once you learn this structure, then you should also know that, as we have already mentioned, that these proteoglycan molecules then aggregate in the form of monomers, but they are not aggregate in the sense that they are crowded with each other, because of they are not crowded. They are in well separated way. They are arranged in a very well mannered way, and this separation is because of the repulsion they feel from each other. So in this way, the different monomers of the proteoglycans are arranged on a linker acidic molecule, hyaluronic acid, and you can see this is one monomer, this is one monomer, this is one monomer, and this is one monomer. Each monomer again in turn is composed of this thread-like structure represent the carbohydrate part, that is GAGs, and then in the center is the core protein. So in this way, these molecules make a meshwork of network in the ECM or wherever they are playing their role. So amino sugar again are very important component of proteoglycans because they are making the GAGs. Their importance can also be seen because of the fact that they are present in many important molecules like antibiotics also, which have their own significance. You all know about that. Then in connective tissues also, because these are adhesive molecules, at least 20% of the glucose is used in the formation of these amino sugars. It shows their importance. So you get the fructose 6-phosphate, which is a precursor of this pathway, which is a precursor for different units which will be used in the formation of proteoglycans. The fructose 6-phosphate then undergoes the amynation process through the amylotransferase enzyme, and the glutamine amino acid is used here. Glutamine is converted into glutamate, and you get the glucosamine 6-phosphate. This glucosamine 6-phosphate that underwent the acetylation process through the acetyl-CoA, and you get this N-acetyl glucosamine 6-phosphate. N-acetyl glucosamine 6-phosphate then adopt two routes, one this one and one this one. In one, you get the N-acetyl manosamine 6-phosphate, and the other route, in the other route, you get the UDP N-acetyl glucosamine. Why UDP? We have been saying it again and again that in the formation of such large molecule, you need lots of energy, and the molecule that these molecules, like glycogen was doing, they get their energy from the UTP. UTP gives rise to the formation of a UTP molecule. Here, that UTP molecule is the UDP N-acetyl glucosamine. This UDP N-acetyl glucosamine then is converted into UDP N-acetyl galactosamine. Now, UDP N-acetyl glucosamine and UDP N-acetyl glucosamine, galactosamine, both of these act as the major units of the proteoglycans. They make the GAGs. While the N-acetyl mannose 6-phosphate is converted into NANA, that is N-acetyl neuramine acid. This is, although NANA is not as frequently used in the formation of proteoglycans, instead it is mainly used in the formation of sialic acid, gangliosides, and glycoproteins. But something very important here, you should note that here the nucleotide that is this the high energy compound is not UDP based. The NANA is not UDP based but CMP based. So perhaps this is the only molecule in our living bodies where CMP acts as the high energy compound for the further utilization of NANA. In other compounds, we have been seeing it was the UDP which was being used, but here it is CMP, which makes it a very specific molecule.